lecture is about hybridoma technology for the production of monoclonal antibodies. To introduce the topic, antibodies are glycoproteins synthesized in the blood against a specific antigen to combat and give immunity. Such antibodies are heterogeneous and are polyclonal antibodies. Therefore, they do not have a specific character. If a specific lymphocyte, after isolation and culture in vitro, becomes capable of producing a single type of antibody which bears specificity against a specific antigen, it is known as a monoclonal antibody. These monoclonal antibodies are derived from a single clone which recognize only one kind of antigen. Monoclonal antibodies are produced against a variety of proteins. They are glycoproteins, glycolipids, nucleic acids, and chemically defined groups linked to protein carriers. The hybridoma technology involves the production of monoclonal antibodies. The term hybridoma is used to fuse cells resulting from the fusion of two types of cells, that is, a lymphocyte and a tumor cell. An antibody producing B lymphocytes, for example, spleen cells of mouse immunized with RBCs from sheep, and a single myeloma cell, which is a bone marrow tumor cell that can be adopted to grow for infinite time in culture. The fused product derived by the ability of the two different types of cells, that is, that which has the ability to produce large amount of pure antibodies as lymphocytes and ability to grow or multiply indefinitely like a tumor cell is called as the hybridoma cell. This picture represents the hybridoma technology. It involves the immunization of a rat with a specific antigen from which we can obtain the B cells and fuse it with the myeloma cells to produce the hybridoma cell. And then the produced hybridoma cells can be selected using the hat selection, from which we can specifically separate out the hybridoma cells and screen them for the antibody of interest. Hence, these antibody-producing hybridoma clones can be multiplied further for the production of monoclonal antibodies, which can be used for the future purpose. The major steps which are involved in the production of monoclonal antibodies are as follows. The first step involves immunization of the rabbit or a rat and extraction of the B lymphocytes. So mainly to obtain the B lymphocytes which are producing antibodies, the rabbit or the rat, which is an animal model, is immunized by repeated injections of a specific antigen, which could be a sheep RBCs. A sample of B cells is extracted from the spleen of the immunized rabbit or rat. The next step involves fusion of the myeloma cell with these obtained B lymphocytes. The extracted B lymphocytes are added to a culture containing myeloma cells which are obtained from bone marrow. The intended result is the formation of the hybridoma cells which are formed by the fusion of B cells and the myeloma cells. This fusion is done using polyethylene glycol, electroporation method, or by using phages. The next step involves the selection of these hybridoma cells. The B lymphocytes contain the HPRT1 gene, which codes for the enzyme hypoxanthine guanine phosphoribosyl transferase. The enzyme HGPRT, involved in the synthesis of nucleotides from hypoxanthine, is present in the culture medium. Therefore, the B cells can grow in a medium containing hypoxanthine aminopterin thymine, that is the HAT medium. But myeloma cells lack the HPRT1 gene, so it does not produce the HGPRT-PTR enzyme, and hence it does not grow in the HAT medium. The myeloma cell fused with another myeloma cell, or those that do not fuse, will die in the HAT medium since they do not utilize the hypoxanthine. Similarly, the B cells which fuse with another B cell or those that do not fuse will die eventually because they do not have the capacity to divide. So only the hybridoma cells, that is the fused cell between the myeloma and the B cell, can survive and divide in the HAT medium. Screening is done to select these hybridoma cells, which are the desired cells for monoclonal antibody production. The next step involves culturing of these hybridoma cells. The selected hybridoma cells are cultured in a suitable medium and are also supplemented with insulin, transferon, ethanol, amine, and other additional hormones for better growth and development. Some of the commonly used culture media for the production of monoclonal antibodies are DMEM, which is Dalbeco's modified Eagle medium, IMDM, HAMS 15, and RPMI 1640 medium. The next step involves inoculation of these hybridoma cells into a suitable host. The hybridoma cells are then injected into a lab animal, which is the host, where it starts to produce the monoclonal antibodies. And these hybridoma cells can be obtained and frozen and stored for future use. The next step involves extraction and purification of the monoclonal antibodies. The monoclonal antibodies, which are produced and obtained from the host animal, is extracted and purified by any of the following methods, that is ion exchange chromatography, antigen affinity chromatography, radial immunoassay, as well as immune precipitation. This picture is again a representation of the entire hybridoma technology, where an immunized rat is used for obtaining the B cells from the spleen, and then they are fused with the myeloma cells. The fusion results in the hybridoma cell, which is selected using the hack medium, and then they are tested for the presence of the specific antibody production types, which are then inserted into a suitable host where the multiple amount of monoclonal antibodies are produced, which can be harvested. The harvested monoclonal antibodies can be stored for the future use. The applications of monoclonal antibodies are as follows. It involves disease diagnosis, where various methods like ELISA, RIA can all be applied to detect various infections and for the diagnosis of various diseases. Then they're used for disease treatment, where a particular type of monoclonal antibodies can be used for specific antigens. There are also different types of monoclonal antibodies can also be used for radial immunodetection and radial immunotherapy for cancer. The other applications involves passive immunization or disease prevention, where monoclonal antibodies-based drugs can be prepared for septic shock, as well as it can be used as a vaccine. And further, detection and purification of biomolecules can also be done, where monoclonal antibodies are very useful in determining the presence or absence of specific proteins through Western blotting technique. Hence, the various applications of monoclonal antibodies in the field of animal biotechnology has found improved applications. Thank you.